So potentially this is our last lecture time. I will certainly be here next Thursday. And if you want to come in and you got questions about anything, come see me. I'll be here. Obviously, if we don't finish stuff, then I'll be here teaching you also. So, but you have everything you're going to need for the next test, all that kind of good stuff. So, there are 4 sheets. If you're not here, or if you're walking in really, really late, that's all right. Come grab one of each of these 4 sheets. Wondering who I'm talking to, I know, for instance, like Jaylee will watch this online when she's out of town with golf this week, so I'm embarrassing her not to be able to complete here to. Alright. I need one of these. Okay, so you got these 4 sheets. These are intended to help you review and also to fill in, just in case there's anything that we have left out that you need. Where do we want to start? Let's go, this one will be the last one, okay? This is like, hey, how do I study for this test? What, you know, that's this one here, okay? Same thing, but let's begin with this one, with the tiniest of print. I'm sorry it is so small, but it is what it is. Okay, so it's really long, skinny. All right, so it's a chart, right, or a graph, or whatever you want to call it. It's got the various phyla. I'm going to do this. You guys have had this before many times, so I don't know that you need to write it down again. You can if you want to, but you know we've got all of life, right? So we divide it into 2, and then over here we've got archaea and bacteria. Over here we've got protists, and we've got fungus, and we've got plants, and we've got animals, right? So here we are, we're dealing with this one. Now I'm going to subdivide this in a major way. Okay. So over here we've got porifera, and then Cnidaria, and then Platyhelminthes. You've got nematoda, and then you have Annelida, and mollusca, and we have Arthropoda, and then we have Echinodermata. And you might want to draw this, at least this bottom part, okay? Echinodermata. And then over here we have Chordata. These are all the animal things. You gotta know these, okay? These terms are gonna be all over this test. You don't have to be able to spell Platyhelminthes necessarily, you're gonna see it, okay? You got to know. What I always used to teach in class was plat means flat. These are flatworms. So I think I would just, well, how am I ever going to remember what that is? Well, think with the P, think an F. Flat. These are flatworms. Porifera sounds like it has what in it? Pores. In fact, that's what the name means, pore bears. So think of some super simple animal with just these holes all through it. Well, that's a sponge. So these are sponges. Cnidaria, that's a little trickier. The key there is it comes from the word cnidocytes, which are stinging cells. So if I think Cnidaria, cnidocytes, I think I can put those 2 together. And if any way I can think of cnidocytes as something that stings, well now I know a jellyfish is the thing that stings, right? Here you are, sir. Again, plat means flat. Nematodes, they're not toads, they're not frogs at all. These are simple little worms, little round worms. Annelida, that's another weird one, but they're cool. So these are, I guess maybe one thing to think about is that from here to here, you're kind of dealing with worms, okay? Pretty low level life. Flatworms, roundworms, earthworms, kind of in that order. These have more than just earthworms though. These do have earthworms. They also have leeches, and they've got stuff like Christmas tree worms. These are the best. Have you ever seen a Christmas tree worm? They live in the ocean. And this is not going to be a good drawing of them, okay? Because I'm just drawing what I can. But it's a worm, and then just all of a sudden at the top, it's got like this Christmas tree looking thing that sticks up. They're crazy cool. And they're in like these bright colors. Don't eat those. If it's a bright color, don't eat it, okay? Like just that is always God's warning to you, this will kill you. Probably have like eye spots or something. But anyway, they're cool. So worms. Flat, round, earth. Okay? Well, that's fun, like flat earth, round earth, people who believe weird things. Alright. Forebears, right? Sponges. Cnidaria, cnidocytes, stinging cells, jellyfish. Nematodes, roundworms, annelids, earthworms, leeches, Christmas tree worms. Mollusca, this word means soft. It's Latin. Soft. What's the softest animal you can think of? It's a slug. Have you ever seen a slug? A slug is a snail without a shell. Like it's so soft that if you just pour some salt on it, it just like into nothing. These have like the softest bodies. Squids, octopus, clams, oysters, snails, slugs. There are others, but we're not worried about them all. We're doing a big overview, right? So mollusks, these guys are soft. Anything that you're seeing at the beach with a shell, that's a mollusk. I mean, unless we're talking about like a crab or but those are different. But the hard shells that you collect and all that, they're mollusks. Arthropods, jointed. And pod means foot, so jointed feet, jointed legs. So these are like insects and crabs, and these guys have exoskeletons. So we're looking at good protection now. Most of these guys have some kind of a shell, or at least really cool camouflage, or they can squirt ink at you. But these guys are we're getting pretty advanced here. We got like brains, and we can problem solve, and we can really respond to stimulus. We can hide. We can change colors, we can squirt ink, like, this is getting good. Arthropods, exoskeletons, that crunchy, that's these guys. So anything that you would ever like step on, a spider, a tick, a mite, a scorpion, don't step on butterflies, but you could. A butterfly, these are arthropods. So these are all your insects, spiders, ticks, mites. Scorpions, insects. One of the coolest things that you might not even know about, like a horseshoe crab. Oh, this is also, we've also got here like crayfish and crabs and lobsters. You ever heard of a horseshoe crab? They're super cool. They kind of look like this. They look like crazy. I mean, this is terrible. I'm making this look like a bad animated version. I don't know. Yeah, that was terrible. I'm going to erase that. That's awful. They're cool looking. They smell though. Like echinoderms. Echino means spiny. And dermata, like you go to a dermatologist, spiny skin. Right? So the spiniest of all the skins that you will ever see belongs to a sea urchin. They are wild, right? But if you were to look at a starfish, they also have little spines on their skin. They're just much smaller. Okay, so spiny skin. And then chordata, chordates, there are a number of things, but there are really three groups here. I don't even need you to know the names of the first two so much, but you've got lancelets, you've got sea squirts, and then you got everything with a backbone. In other words, these two things are chordates, but they don't have a backbone. Everything else has a backbone. So flat meant flat. These things are round. These things have cnidocytes, stinging cells. These things are, the name literally means pore bearer. They have pores. Okay? They're filter feeders. These guys are hunters. They have stinging cells and these long things, and they not very well controlled though. Jellyfish can't they can do this, right? They can pulse a little bit, but they don't swim. They don't go to a destination on purpose. They go wherever the ocean takes them. So their hunting is, so to speak, is fairly random. Like if we touch you, we kill you. But if we don't touch you, you're safe. We can't go get you. I remember playing the Super Mario Brothers video game when I was a kid, and they'd always have the jellyfish, and they would hunt you out. That's not how it works in real life. Flat means flat. Nematodes. Okay. So where were we? What were we looking at here? Which sheet did I tell you on? The long skinny one? Okay. So these are all of the animal phyla, right? With a big overview, you got to know the names, or at least be able to recognize them. And you're certainly going to have to know some of these characteristics about them. It's kind of a big deal. There's a lot you'll need to study. Do you understand this? You really, you got to be able to reproduce this chart. Okay, I'm just telling you right now. You must be able to reproduce this chart. Which means you got to understand it. If you understand it, it's easy. It'll take you no time. But if you don't understand it, forget it. So we have some terms on here like blastula. A blastula is, you got an egg and you got a sperm, and they come together and they fuse, and then it begins to divide, right? And it forms a big hollow ball of cells called a blastula. If you did not know that, write that down. A blastula is a big hollow ball of cells. Well, all animals have that. All of them. So for blastula, it's just everybody. And for most, but not all, something happens early in its development and it forms this rudimentary gut. This, like in you and I, this thing right here folds in and another one folds in from the other side, and that forms what we call your alimentary canal, your digestive tract. This end would become your rectum, that end would become your mouth, right? But your gut forms from this first pouch that begins to fold in. What we're looking at here is what happens basically in an embryo. Within hours of an egg and a sperm fusing, this starts to happen and your gut begins to form, okay? So this is like the blastula. When we start folding in, this is a gastrula. Gastro means stomach. Do you know that? It's more of that Greek stuff, right? So gastro or gastru, that is stomach. Well, here's an easy way to know which things here have it and which things don't. This does not. It has no true tissues. See, this is going to give rise to at least a rudimentary stomach and digestive situation with true tissues forming, rudimentary organs forming. These don't have that. These have no tissues, no true tissues. They're not. The idea of a tissue, if you don't remember, is that you've got cells that are specialized. They do like one job. And other cells do their job, and other cells do their job. Over here, every cell, potentially can do every job. They don't have true tissues. So this porifera has no gastrula. So that's one way of thinking about it. Another is just, remember, when I'm giving this to you, I'm giving it to you in order of increasing complexity. So the simplest things lack the complex things. Everybody's got this, but the first thing on there does not have a gastrula. Okay? Now, a jellyfish kind of looks like that, doesn't it? It's like that with some tentacles hanging off, doesn't it? And so you go like, all right, here's my jellyfish, and I got, some stuff hanging down. Well, that's because that's basically all this is. It's got a little rudimentary stomach, mouth, gut thing. It's a gut, okay? It doesn't have a mouth and a stomach separately. It's just got like a gut cavity. And so these tentacles might sting something and bring it up towards the gut. It's going to release enzymes. It's going to digest that food in the gut, but that's also open to the water, right? So this is not the most efficient thing you've ever seen. These have true tissues, okay? So this has a blastula, and so does everything else from then on, okay? This doesn't, everybody else does. Okay, now, from this blastula, we begin to have some basic tissue types that form. Okay, you're going to have endoderm and ectoderm at the very least. This is going to form endoderm, and this is going to be ectoderm. How many layers is that? How many layers is that? Two. My 2-year-old says 2 better than y'all say 2. Benjamin, how old are you? 2! He's very excited to say it. Come on. All right. So Greek prefix for 2 is di. Dip, as in like dip, low. Diploblast. So if you look on there, it says diploblast. A diploblast is an organism that has only 2 types of tissues, an endoderm and an ectoderm. Well, that's these guys. That's all they have. Just an endoderm and ectoderm. They don't have a complete tube going through. They don't have a separate mouth and anus. They surely don't have mesoderm, anything between the two, okay? So your diploblasts are your cnidaria and your cnidaria only. So there's no such thing as a monoblast, all right? You either have this gut forming or you don't. So you either form a gastrula or you only have a blastula. There's no monoblast. But from the gut forming, we can have diploblast or we can have triploblast. We only have one diploblast. Everything else here has a triploblast. 3 germ layers, 3 layers, distinct layers of tissue formation, okay? And so what happens with everybody else is as this embryo, this zygote, whatever, begins to develop, right? this becomes much more of a serious thing, and we're starting to take on a different shape. This is going to form, right here, is all going to form my endoderm, right? Okay. This out here is all going to form my ectoderm. And now in between, I'm going to form another layer of tissue called mesoderm. So, everything from Platyhelminthes on has 3 layers. With me there? Everything else, from flatworms on, has 3 layers. Now, my flatworms, they have some advantages over being a cnidaria, but they're not as cool as you. They don't have a mouth and an anus separately. They don't have a complete canal. I drew these for you a while back, and they're fun to draw. They look like this. A lot of things I tell you, I'm going to draw this, and it's not going to be very good. This is exactly what these things look like. You look at one under a microscope, this is exactly what they look like. They've got these eye spots. They're not eyes, but they can't detect colors, shapes, stuff like that. But they can detect, like, oh, there's light or there's not light. And so they can go to the light or away from the light, depending on what their needs are. But the most distinguishing feature about them all is this gut. The gut runs from the mouth all the way down to the tail, because they don't have a separate anus, and they don't have a stomach. They don't have digestive organs and things like that. They rely on simple diffusion. They just get the food in, and it goes everywhere, and the poop and whatever waste materials, it goes right out through the same thing. It's like basically just having a trash can in the middle of your body. They just spit it back out. Why am I talking about these? Because these are triploblasts. They have the 3 germ layers. But as the body, I'm going to now draw like a cross segment. Okay, what would this look like if I cut it crosswise, especially as it was developing? And I got to erase a little bit here. So if I were to cut it crosswise, okay, I have got my ectoderm on the outside where I'd expect it to be. Yeah, I did that wrong, didn't I? That should have been green. Let me stay consistent. Okay, so I've got my ectoderm. I've got my endoderm. Then I've got my mesoderm. Cross section of them looks like this: ecto, meso, endoderm. This is the gut, right? This is that. Other critters look different though. If I were to go from a flatworm to a roundworm, things look largely similar. This is flatworm, right? Now, if I were to do a roundworm, ectoderm, endoderm, and now I go to draw my mesoderm. Notice there's a gap here, okay? I'll have another thing. This gap is what we call a pseudocoelom. Pseudocoelom. And there's a third body plan. It's the one that you have. Third body plan looks like this. Got my ectoderm. Got my endoderm. I've got my mesoderm. And I've got these holes in my mesoderm. Now these holes are what we call a coelom. So flatworms do not have coeloms, okay? They're what we call that means without, acoelomates. These have something that looks like a coelom. I've got this fluid-filled body cavity, but this fluid-filled body cavity is 100% surrounded, or excuse me, is 50% surrounded by mesoderm and 50% surrounded by endoderm, whereas over here it's 100% surrounded by mesoderm. Okay, so this is a coelomate, this is a pseudocoelomate. Everything else is a coelomate. Well, let's look up here real quick, okay, and I'll put some pieces together for you. So, platyhelminthes, these are acoelomates. These guys are pseudocoelomates, false coelomates. And these guys are true coelomates. And so is everything else going that direction, okay? What we're looking here is embryonic development. This one only has a blastula, no gastrula. This one has a gastrula that's going to form, this gut, right? But it doesn't. Because all it does is form a gut and not like a tube. We're not, it's only got 2 layers. We have mesoderm we have ectoderm and endoderm. But here we have 3 layers. When I look at these 3 layers, I notice these 3 layers are simpler than these other sets of 3 layers. These have a fluid-filled cavity that this does not have, okay? I call that fluid-filled cavity a coelom. These don't have it. These do, and these do, but they're not the same. They're not the same at all. This one is only half surrounded by mesoderm. This one is completely surrounded by mesoderm. This completely lines that endoderm also, and this does not. Now the question is, what is this? Other than saying it's a coelom, who cares? But what does a coelom do? A coelom is where your organs develop. It's where your kidneys, it's where your intestines, your pancreas, your liver, all those things develop inside of this. Does that make sense? Okay, so these don't have one quite like that. They have this, and this is cool. They actually can use hydrostatic pressure to use this almost like a skeleton to contract their bodies. but they don't do what you do. And so we're getting more complex as we move on, and we can see that complexity at the embryonic level, how these things develop, okay? We can see things at the embryonic level that we can't see at any other level. We'll cover that more in a minute. So, Porphyra, Cnidaria, Platyhelminthes, Nematoda, annelids are the first ones that are true coelomates. They have all this, okay? Going back to our chart here. Blastula, gastrula, diploblast, triploblast, acoelomate, pseudocoelomate, true coelomate. Now we get into things like protostome, deuterostome, spiral cleavage, and radial cleavage, and symmetry. Okay, so stome, stomata means mouth. Okay? Remember, I said here that for you, this first hole that forms your anus, your rectum, and the second hole forms your mouth. That's true of you, and that's true of echinoderms. But for everything else, for annelids, for mollusks, for arthropods, this first hole becomes their mouth, okay? Protostome means first mouth. The first hole, first indentation forms the mouth, okay? So, annelid, mollusca, arthropoda, these guys are all first mouths. Protostome means first mouth. Okay. That's something you can only see at the embryonic level. You would never know that, oh, the first hole there, their mouth came from this as opposed to from that. You'd never know that once you saw it, right? But if you watch how it develops and you just track where this is going, you see the mouth come on this end for an earthworm, for a mollusk, for an ant, for an insect, but it's very different for you. If you watch a human or a chimpanzee or anything with a backbone, and you watch it develop, you notice that this end becomes the rectum, and the second one becomes the mouth. So second mouth makes you a deuterostome. This is, again, we're going back to the Greek. That just sometimes in this class you just have to know. Deutero means second. Deuterostome is second mouth. I'm sure I told you this before, but in the Bible, the book of Deuteronomy, deutero means second. Onomy or nomos refers to law, so it's second law. They call it that because you get the Ten Commandments the first time in Exodus, you get it the second time in Deuteronomy. It's there twice. So second law. All right. So we have protostomes, we have deuterostomes. Again, this is at the embryonic level. We're following like this ball of cells as it develops and grows. There are distinct patterns that God has given His creation. And it's consistent, it's organized, it's brilliant. Now, some other things happen with our protostomes and our deuterostomes. One thing is that as look at the blastula again, we have spiral and we have radial cleavage. So in spiral cleavage, these cells become kind of offset. They spiral away from each other. They've got like a directionality to them. Let's see if I can show this or not. It's got like a definite spiral to their development, if that makes sense, okay? And spiral cleavage, kind of looks like an Easter egg. Spiral cleavage goes along with your protostomes. They go together. Okay. Deuterostomes go with what we call radial cleavage. They just simply radiate out from each other. They just kind of look like this. It's kind of like basketball stuff. Makes sense? So, if you understand orders of complexity as we move forward. If you know what a stome is, that's a mouth. If you know what a gastrula and a blastula are, if you know mesoderm, an endoderm, an ectoderm, if you know what a coelom is, acoelomate, pseudocoelomate, true coelomate, then this chart becomes actually pretty obvious, right? Well, of course, everything has a blastula, because that's where it all starts. But then, okay, Cnidaria, they have a gastrula, they have a primitive gut, but sponges don't, so I'm going to leave that off there. All right, well, all the things that have a gut, only one of them only has 2 germ layers, and that's the Cnidaria, right? The things that do this. They only have an endoderm and an ectoderm, so they're the only diploblasts. Which means everything else on here has to be a triploblast. All right, now out of my triploblasts from here on, flatworms are the only acoelomates, nematodes are the only pseudocoelomates, everything else is a true coelomate, so that fills out the rest of it. Now, I'm looking at protostome, now I'm looking only at the things that have triploblasts, or that are coelomates rather, and I'm going, all right, which hole forms the mouth? Is it the first one or is it the second one? Well, for the first 3 here, it's the first one. For the second, it's the second one. Now, and corresponding with that is radial and spiral cleavage, okay? They correlate 100%. The last thing up here is symmetry. Okay. A sponge, folks, could look like, I mean, it could just look like almost anything. Would you be able to draw a line of symmetry in that anywhere? No. So these are asymmetrical. A Cnidarian, if I were to turn it upside down, it tends to look something like this. Or maybe it looks like this. Yeah. Can I draw a line of symmetry there? Sure, I could draw them almost anywhere, right? There's radial symmetry. And I could draw a ton of them, couldn't I? I could go like, all right, there's symmetry, there, I could turn it into a pie. That's radial symmetry. So this is asymmetrical, this is radially symmetrical. Everything else has bilateral symmetry at some point. A flatworm? We drew a flatworm, right? How many lines of symmetry do I have? 1. So everything, so the symmetry is easy. It's asymmetry, it's radial symmetry, and everything else has bilateral symmetry with one exception, okay? And that's my starfish and stuff here. Can I draw lines of symmetry here? Yeah, how many? 5. The thing about these is they develop this later. They don't start off with 5 planes of symmetry. They start off with just 1 plane of symmetry. They begin bilaterally symmetrical, and then as they develop, they turn into this, what we call pentaradial symmetry. So primary symmetry for them is bilateral. Then they have secondary pentaradial symmetry, okay? You got to know that. That's on here, okay? So these guys have secondary pentaradial symmetry. Make sense? Hopefully, at least for now. Hopefully next Tuesday also. So secondary pentaradial symmetry. All right. And everything else just has bilateral symmetry. You can draw one single line of symmetry. There's one other term, it's not on here, but it is a major term that you need to understand, and it's cephalization. Cephalization looks like this. We have an organism called a cephalopod. Cephalo means one thing, pod. Do you know what pod means? Do you remember? Head up there. Arthropod, foot. Cephalopod means head, foot. Cephalopods are your squids and your nautiluses and your octopi. Cephalo means head, so it's a head, foot. So cephalization is the idea that you're going to have a distinct head. What it really means is you're going to concentrate your sensory organs, your your brain power, your everything else in your anterior end. Right? Think about it. All your, like, if you want to look at something, you don't stick your hand out and like look at it, right? You don't taste with your toe. Like, all of your sensory stuff you keep in one spot. And that begins right here with the flatworms. These eye spots, they're the first ones to show, like, cnidaria, their nervous tissues throughout their whole body, just randomly, randomly or evenly distributed. These guys don't have it at all. Already at the flatworm level, we've got our sensory stuff in the anterior end. Okay, so it's an important concept. Your life would be very weird if you didn't live like that. Okay, so that is this chart. You got to be able to recreate that. Anything on here, any which way, is going to be fair game. Okay, and the vocabulary that we've covered is fair game. Yes, ma'am.
Are we going to have to know how to draw them?
No, you don't have to draw nothing. Yeah. Not in our class. Okay, so what's next? What are we going to look at next? Let's go this one. It's the one that says blastula, gastrula, diploblast, mesoglea, triploblast, acylomate, silicylomate, coelom. Okay. So what is a blastula? I realize you don't actually have room on here to write this. I didn't think through that very well. I'm sorry.
Hollow ball cells.
Huh?
Big hollow ball.
Yeah, it's a ball of embryonic cells. Hollow ball of embryonic cells is a blastula. If I can take a second, I can remake this to where you guys can actually do something with it. It'd probably be worth it for you, right? Printing, grab it, and you can actually write it on. All right. So what is it? Blastula, hollow ball of embryonic cells. By the way, I'm going to erase this. Gastrula, what is it? Huh? Ball that's forming a pocket, right? It's basically a blastula that's forming a gut. That's the difference. A gastrula is basically a blastula that's forming a gut. You understand it like that, that'll help a lot. What's the next word on there? Diploblast. What does that mean?
2 layers.
Yeah, 2 distinct tissue layers, right? And what's next? Mesoglea. Okay. So mesoglea is the space between those layers in a diploblast. Or the fluid, maybe, between the layers in the diploblast. If you cut a jellyfish, you know what it does not do? It does not bleed. If you cut an earthworm, it bleeds. If you cut a jellyfish, it does not bleed. It has no blood. It has no means of moving materials around, no vascular system. But in the mesoglea, stuff is free to move. Nutrients, waste, gases, etc. Okay. Next up is triploblast. We know that those are 3. And do you know what the germ layers are? What are the germ layers in a diploblast? What are they? Endo and ecto. You might make note of that. What are they in a triploblast? Endo, ecto, and meso. Good. Acoelom, what is it? No coelom. Good. What's a pseudocoelom? Well, the name means false coelom, but what is it? That's a fluid-filled body cavity surrounded by, on one side by mesoderm and one side by endoderm. Fluid-filled body cavity. Surrounded by meso and endoderm. And then a coelom is a fluid-filled body cavity surrounded only by mesoderm.
Like you said with the
Pseudocoelom?
yeah. Can you say what?
It's up here. So fluid-filled body cavity surrounded by mesoderm and endoderm. And then a true coelom is a fluid-filled body cavity surrounded only by mesoderm. So that's what it is. Next thing, like, what does it come from? What does a blastula form from? From the zygote.
Division of cells.
From the zygote. What does the gastrula come from?
blastula.
From the blastula. Good. Diploblast, mesoglea, triploblast. Where do they come from?
Yeah.
The gastrula.
All three.
Yeah. This is just following embryonic development. One thing becomes another, becomes another, becomes another, right? The gastrula will either form a diploblast or a triploblast. If it's a diploblast, the mesoglea is also going to come from that blastula, right? All right. Then a triploblast, where does it come from? I think we already put it, right? It comes from the gastrula. Acelome. You just put an X through that. We're not worried about that. Pseudocoelom, where does it come from? What two layers? Endo and meso. Endo and meso. And coelom, you put meso. Huh? What did I say? No, coelom is meso. True coelom is surrounded exclusively by mesoderm. Otherwise, it would be inside of your elementary canal. You know what I mean? Okay. All right. What does it become? What does a blastula become? Hmm? A gastrula. Or else, I guess a sponge, but we're not worried about them right now, right? What does a gastrula become?
Either diploblast or a triple.
Becomes a gut
Okay.
or a stomach or a digestive tract, depending on how, you know.
Digestive tract.
Yeah, just digestive tract or a gut. Remember, if it's a Cnidaria, if it's one of these jellyfish, then all it has is a gut.
Okay.
Same thing, really, for an acoelomate, right? Okay. What does it become? Diploblast. You can go ahead and write endoderm and ectoderm. Triploblast, you can write endo, ecto, and mesoderm.
Use which one?
Triploblast. We're just listing out the germ layers. Coelom, acoelom. Put an X. Doesn't do nothing. It's not there. Pseudocoelom, you can write this. Hydro, static, skeleton. It's not a true skeleton, it doesn't have bones, but by putting pressure around this and contracting against the fluid that's inside of it, it's able to have muscles and things contract and attach and move around it. It's not smooth. A roundworm, they move like somebody's having a seizure. I mean, it's wild watching them move. It's not pretty. But they can only do what they do because of the pseudocoelom that's forming a hydrostatic skeleton. What does it become for a coelomate? It becomes your internal organs. What does it do? You can skip down to mesoglea. You can put X's through the first three. Okay. Mesoglea, you can say that it transports materials. Then you can put X's on triple blast and acoelom. So take you down to pseudocoelom. Do me a favor when you're down to pseudocoelom, when you're ready for pseudocoelom, look your eyes so I know I don't want to skip anything. I'll be teaching next class, so I would come. It'll help if you study in advance, but I would come. Pseudocoelom. So what does it do? Well, it helps with movement, right? I'd write that down, helps with movement. All right, that brings us to,
Could we put an X on the last one?
sure, yeah. So it brings us to this one. It has all these listed in order, running down. It's just given a ton of characteristics about them. So these are the things that you must know. Like, you must know them. Because if you were to look at the other thing that you have, take a look at the top. It says, what phyla does it belong to? And then it names them all. And then it'll say things like, somewhere on there it'll say sponges. Well, you must know that a sponge is porifera. The next thing on this one, it says pore bearer. Well, pore bearer is on here too. So when you see pore bearer, you must go, well, that's a porifera. It says no symmetry, asymmetrical. So when you see asymmetry on here, and it's there, it's like the third one down, then you got to go, okay, that's a porifera. No tissues or organs. Do I have that on here? I hope I do.
You have no tissues to what?
No tissues, great, that's how important it is. All right,
What?
so, yeah. My editor missed that. So on all of those terms, these are what we'd call distinctives. These are things that stick out. They're true of this organism, okay? There's nothing else in God's creation, as far as an animal, that does not have true tissues. There's nothing else that has these pores and filter feeds like this. There's nothing else that lacks symmetry. It's these guys and these guys only. So if I give you those identifying characteristics, you've got to be able to tell me, hey, that's a sponge. Now, that might just sound like a mean game to play on a test, but by making you know these, and to know that these are true only of sponges, you're going to necessarily learn all these things about a sponge. A sponge has no symmetry, has pores, filter feeds, it has no true tissues, it's in the phylum Porifera, it's the simplest of animals. So there is method to this. This is not just me being nitpicky and a jerk. All right, we're on short time, so how do I get you to get all this in, and in a way that's ultimately going to be fair, because you know how I'm going to test you on it. So that's where this is coming from. I hope that'll ease a little bit of what might feel weird to you. But you must know this, okay? Here's what you don't have to do. I'm not going to. It would be fair if I did, but I'm not, because I don't have time to read essays. But I could easily give you a little, short answer, like, tell me about sponges.
You don't want to do that.
I'm not. No, I don't want to read that.
Mhm.
No, instead I'm going to make you fill out charts that will. So you don't have to have it for recall, every single one, but you have to have it for recognition. Okay? You got to recognize, when you see it, you've got to know it. Okay? Nidaria. Named for cnidocytes. These are stinging cells. This is why when you touch one, like it's, I hate that, right? I've never been stung with it, but it's always a chance. So they're named for cnidocyte stinging cells. They have muscles and a rudimentary nervous tissue, but nothing like a brain or a heart, right? They're not the simplest animal, but they're the second simplest animal. They don't have hearts, they don't have blood, they don't have stomachs, they don't have livers and intestines and gut, they just have a gut and some stinging cells, and they can kind of sense light. And they can detect when they've been touched or when they touch, and they can contract things. Like sponges don't do that. So these are definitely much better advanced, much better equipped than a sponge is. They float with the currents. They undergo 2 distinct phases of life. Go like this. Ready? Come on. Come on. Go like this. Everybody put your hand up. Nice and loose. Loose, loose. Now, pulse. What is this? What are we mimicking? A jellyfish. So these long tentacles. No, stay here. Stay with me. Come on. I don't make you do that many goofy things. So, Greek mythological gorgon. Do you know what a gorgon is? Oh, man. There were 3 gorgons. They were sisters. And is it Perseus that had to go and kill them? And if you looked at him, he turned to stone. Do you know the one's name? Her name is? Medusa. So this is the Medusa phase. Literally, that's what it's called scientifically, okay? So this is Medusa. Medusas are jellyfish. And jellyfish are, there are other cnidarians other than jellyfish, but you know these. The other is go like this, polyp. So if it's upside down doing this, it's a Medusa. If it's up, I guess it depends on upside down, right side. Those are relative terms. If it's doing this, if it's dangling, that's a Medusa. If it's like this, like a vase or something like that, it's a polyp. Okay? All of them have both phases. Okay? The only difference is this or this. So you might think, well, you're stupid, Mr. Douglas, for making us do this. But like, you should tactilely now have like, oh, this is a Medusa, this is a jellyfish, and that's the other thing. It's called a polyp. Okay? So hopefully that helped. They undergo 2 distinct phases of life, the Medusa and the polyp. These include jellyfish, hydrozoa, corals, and sea anemones. And you must know all 4 of those, at least to know what they are. So, a jellyfish, hydrozoa is like the best example of a polyp. They anchor to like a rock. They're small. These are not very big, okay? But then they have like these, and they're doing what they're doing what a jellyfish does, but they're just staying still doing it. That's not a good drawing of one. But anyway, so they have the stinging cells, and if something comes near them, they'll sting it and capture it and eat it. Okay, so that's a hydrozoa. Hydro means water, zoa means animal, so it's a little water animal. And then sea anemones and corals. You know what corals are, right? Right?
Yeah.
Did you know corals are alive?
Mhm.
Okay. They eat. Stuff comes near them, they sting it, and then they eat it. And they just have this little simple gut, right? Just a gastrula, that's all they have. They only have 2 germ layers. They sting it, they bring it in, they eat it, they spit it back out. That's it. Okay? A sea anemone is very similar. The name for these guys is, oh my goodness. Hello. Is it anthozoa or is it angiozoa? Here, it means flower plant. Have you ever seen a sea anemone? Oh, they're beautiful. Let me show you a picture.
Yeah.
This is worth.
Like with hairs and stuff.
Oh man, they're gorgeous. Come in all kinds of different colors. I have to show you a sea anemone. So sea anemones, corals, hydrozoa. I probably won't give you hydrozoa, but I'm going to want you to know the sea anemones because they're like my favorite. All right, so these guys are cnidarians. Platyhelminthes. Plat means. Say it again. Plat means flat. Help yourselves out. They have a mouth but no anus. Rudimentary gut that spreads through all their body in order to distribute nutrients. So if I said, hey, what critter has a gut that extends throughout its entire body, you would say flatworm, and that would be a fair question. Okay. Oh goodness, they have eye spots. They can sense light. They're the simplest organism to have cephalization. They're also the first ones to have bilateral symmetry. Is that on there? I didn't list it on there. So they have cephalization. They have eye spots, cerebral ganglia. It's not a brain, okay? Ganglia is not. You have ganglia all over your body, by the way. You only have one brain, but you have these little centers of like intense nerve activity called ganglia, and it functions like a little tiny like microprocessor that's then going to send other stuff to the brain. So that's
The nerves?
yeah. You get ganglia throughout your body. It's cool. They're nerves, nervous tissue. Nothing like a brain, but they do help to instantly respond and do things and coordinate.
That's how we feel things in our hand.
Stuff like that, yeah. So it's more than just a nerve, though. It's concentrations, masses. All right. Nematodes, common in soils, can be important parasites of humans and other animals, such as pinworms and hookworms. You and I were talking one day about Bilharzia. That's these guys. Jonathan Bilhars. Okay. Found in soils and waters, they have a pseudocoelom. That's probably the number one thing you must know about nematodes. They are your pseudocoelomates, which lines their alimentary canal. That's the same as a gastrovascular tract or anyway, digestive tract. They have providing a hydrostatic skeleton against which muscles can contract. That's what it's good for. Then we come to annelids. These are your earthworms and your leeches and your Christmas tree worms. Okay. These are the first of the coelomates. You must know that. Guaranteed that's going to be on there. These are the simplest first of the coelomates. They do have true tissues and organs. They have advanced cephalization. Like, it's all happening in the head for them, okay? Everything's happening up there for them. What else? They can, most are hermaphrodites. So earthworm sex, you ready to see it?
I think you showed this to us once.
Did I? Okay. So I over exaggerated this part right here. They have this spot that a lot of, especially when they're ready to reproduce, this will turn like red, or at least fairly bright. A lot of people think of it like a Band-Aid. It kind of looks like a Band-Aid on them. So this is where the eggs and the sperm are. Both of them together, the same spot. Both. Like, this thing is not a he, it's not a she, it's an it. And so you put another one, and they'll kind of like entwine their tails or something like that, and all they do is like rub that spot, and then they'll both get pregnant. Now you know. Okay, so they're hermaphrodites. They have complete alimentary canals, so distinct mouth and anus. The tube runs all the way through. They have like crop and gizzard and like, because these are coelomates, right? So they have these internal organs that develop inside of a coelom, and they're unique and they're distinct. So it's not just like a tube running through, but now this tube has organs attached to it and associated with it for mechanically grinding food, for putting enzymes in, for absorbing food. It's a whole thing, okay? They're the first ones to have that. You think about your digestive tract, right? You've got like the mouth, the esophagus, the stomach. You've got your duodenum, your ileum, your jejunum. You've got your upper and your lower intestines. You've got your, you got the whole deal, right? And then you've got other organs that contribute to that, like your pancreas, your liver, and all that. Well, they don't have everything you have, but they have stuff of their own. Like you don't have a crop or a gizzard, but they do. So they've got organs that are functioning there. Well, those organs had to develop inside of the coelom. These are the first of the coelomates. So of course they're more advanced than everything that was before them, but they're the first coelomates. They're not, they're not as advanced as you. All right, next up is, they have metamerization. That's a fun word. Metamerization. Any biology book ever will tell you that about them. So if you ever look at these guys, you'll see like these lines. You know how you've seen those lines on them? Like what's inside of this line is an exact copy of what's up inside of that one, of that one, of that one, of that one, of that. Like it's like they were put together with Legos. Like little round Lego blocks. Just it's called metamerization. It's the idea of a repeating body unit. So each body unit in this section is identical, and they just stack them. And that's how they work. So that's cool. Mollusca. Bivalves, snails, squids, bivalves. So bi means two, right? So stuff that looks like this, right? Oyster, clam, things like that. Have you ever seen one of these that has a hole in it? Like you're looking at a shell, maybe. And there might be a little pin-sized hole right there. Next time you go to the beach, look at these. Look at your shells, and you will see this in shells all the time. This is because you have mollusk on mollusk, just carnage happening. So these guys do not have what's called a radula, but all the rest of them have a radula. So what happened to something like this was that one of these snails came up, and it's got a shell on it, right? Like something like this kind of deal. I just drew a butterfly, didn't I? Didn't do very good on that. But they've got this shell, right, that they can crawl into and protect themselves. One of these guys came up to one of these, and this thing went, not today, and it closed itself up. This guy said, I have a solution. I have a radula. It's a mouth part that functions like a drill made out of sandpaper. It just grinds back and forth until it grinds through the shell. And then it can just stick its stuff in and start, and open it up and eat it. So it has a radula. I'm going to draw a radula like this. Not really what it looks like, but you get the idea. So radulas are rad. So these guys can literally drill through the shells of bivalves and get them to open up. So that's cool. Mollusks. The name means soft. These have all of them have a muscular foot, which is essential for their movement. It's just filled with muscle and it can contract. And it is the biggest thing about them, and it is soft, which is why their name means soft, hence mollusca from the Latin. Not as many Latin things so far, right? Mostly Greek. They show advanced cephalization, so a head region, with true eyes, brains, and other sensory organs located in the head. They can vary from simple filter feeding, that's all these guys do. All these guys ever have to do is stick up like a tube. Like they don't even have to open all the way up, just enough to get a tube out. They can bury themselves in the sand and eat. Bury themselves in the sand, stick this little tube up outside the sand, draw the water in, filter out anything in it that they want, and get rid of the rest. And they can eat and drink and whatever, poop, just, simple as could be. They can vary from simple filter feeding clams to octopi that can solve, octopi can get out of mazes and puzzles. They can do puzzles, and you can put them on a checkerboard, and they will change the top surface of their skin to match the color and pattern of the checkerboard below. They're amazing. Most have a radula for drilling into shells. All have a visceral mass. Visceral means like deep inside. It's another one of those Greek things. It's like the stuff inside, so they've got a big mass inside. What else do you need to know here? Mantle. Mantle is like this area that usually it might be the part that makes their shell. It's where they're going to breathe from. And a nervous system. And if we had more time, you would have to be able to draw one and all this kind of stuff. We don't have time for all that. What time do I have today, by the way?
Minus 7 minutes.
Okay. We'll do this last one. We'll do arthropods. And we'll
You should have been done 7 minutes ago.
Oh, really?
Yeah.
Oh, I was wrong. Okay. Well, Lord bless us all. You should have said something. So, all right. So we'll pick up with arthropods next time, and then this kind of big review sheets, right? And when we're done with that, if you want to go, you can go. If you want to stay and pick my brain or ask questions or get clarity, you can do that. And then you're, yeah. Okay. Y'all have a good one.